Hi everybody, welcome to my YouTube channel. So today we are going to learn about basics genetics. It's a continuation. So in real life, we see that most biochemical pathways have several steps. So let's just look into one pathway, an example of pathway. Let's say we've got a precursor here, precursor Q, and then got precursor R and precursor S. So these are stages. Let's say this is the first molecule HQ, and then the molecule is modified, is goes to R, and then it's modified, it goes to S, and then it gives us the final product. Maybe it's red pigment. So as we have said in the introduction lecture, we said genes, let's say gene A, they affect these biochemical reactions. We say enzyme A is the one which catalyzes this pathway at this stage, and then we have gene B. We get enzyme B, which catalyzes at this pathway at this stage. Let's say stage 1, stage 2, stage 3. This is a biochemical pathway which is happening. Let's say like respiration or something different. We got on to gene C, got enzyme C, which catalyzes until we end up at the red pigment. So what you realize is that this biochemical pathway has been three genes have played a role in this pathway. So that means if there is a mutation or any destruction of any of these genes, it's going to affect our result here at the pigment. So mostly our biochemical reactions are affected by many genes. Okay, let's go forward. So we have what are called chromosomes in the cells, in the nucleus. So these are very long string-like molecules where genes are aligned. So we are saying they are string-like molecules and genes are aligned. Let's say gene here, gene here, gene here. So okay, let's go on further. It is a molecular structure made up of histones and DNA. So this string-like structure is made up of histone. Histone is a protein, and then DNA, these are deoxyribonucleic acid. So we'll go on further. we look at that, we see that cells of higher organisms usually contain two copies of chromosomes. So cells of higher organisms, they usually contain two chromosomes. Let's say this is our chromosome. And these chromosomes are called homologous chromosomes. This is because the chromosomes carry the same set of genes in the same sequence, although the alleles may not be identical. So we are saying these chromosomes, let's say they have got an allele here, an allele here, in the same sequences, and then an allele here, an allele here. Let's say this is red pigment, and this is the now allele of the red pigment, and let's say this is for wrinkled seed, and this is for round seed. So actually, this is what we are just saying, that the genes are aligned in the same sequence. Okay. So cells or organisms that possess all homologous copies of each of its chromosomes, they are said to be diploid. So these are, for example, the somatic cells of the body. And then we also have single copy of each chromosome. So this is the 2n which you are saying is a double copy of the chromosomes, the homologous chromosomes. And here we have a single copy of each chromosome. There we get a single copy. So this is called a haploid or N chromosome, because we'll be having only one allele. Let's say R here for color. So we have what is called a genotype. This is the genetic makeup of a of two chromosomes. Let's say we have these chromosomes, and we have red for the red pigment and R. So this the R and the capital letter R and the small letter R is what we are calling the genotype, right? Because we are saying on one chromosome we got an R allele which is dominant, and on the other chromosome we have an R allele which is a null allele. So we also have what is called a phenotype. This is the outward characteristics of an organism. This is the outward characteristic of the genotype. So let's say we have an allele here, R and RR, and then we see that the result is a red flower. So the red color will be the phenotype. So Epistas- Epistatic. This is one gene masking the effect of another. So let's look into that. Alleles. We have talked about alleles and we said alleles are alternatives of the same gene. So two identical alleles, they are called homozygous. You see R in R or R in R. These are homozygous. So two different alleles, they are called heterozygous. For example, R and yeah, R and R. So what we are going to talk about next is the dominant allele. Let's say we have our phenotype. Our phenotype red and white. And then we go to the genotype. Our genotype, let's say here is homozygous dominant. And this is because the white color from the beginning, we said it is a result of a null allele, so it will be homozygous recessive. So go on to the gametes. The gametes now is the n number, which we said is the haploid. So we are going to have this is one chromosome and this the other chromosome. So this is the allele in one chromosome and the allele in another chromosome. And here we have got R, small letter R and small letter R. So we are going to, we then cross these organisms. They are going to, there is going to be fertilization. So during fertilization, we are going to see the one chromosome from the side. Let's say the one sperm will bring the R, the capital letter R, and this one will bring the small letter R. So we have in R small capital letter R and small letter R combination. And then we also have this combination which is the same in this combination which is the same, and we will also have this combination. So these all are the same result. So we are going to have this kind of color. So we are going to have this kind of genotype. So a dominant allele, what we are now going to talk about, is when two different alleles are present on one may dominate the situation. For example, in this situation, the capital letter R is representing a dominant allele and the small letter r is representing a recessive allele. So a recessive allele, the allele is the allele whose properties are masked. So that means if we have an R allele representing the red color and the small letter R allele representing the recessive color, which when combined together they form a white. That means this capital letter R is going to dominate the recessive R. So that means all this F1 generation is going to be a red flower. So we go on to talk about what is partial dominance. So partial dominance is when a single good copy of a gene gives results that are recognizable but not the same as the two good copies. So let's have an example. Let's say this is R and you say this is the wild type and it's dominant. And then we have the R, small letter R, which is recessive and it's a nile allele. That means it doesn't produce the enzyme. And then we have, let's say, R 0.5. This one produces 50%, is 50% expressed. So we are going to say that when it's 50% expressed, that's when we say it's partial dominance. So on this word type, we can say it's 100% expressed. This is 0% expressed. So we can see that if the expression is greater than or equal to 100%, we are going to have a red color, or a red pigment or red flower at the result. So if we have R and dominant R and partial dominant R, this is 150, we are going to have a red color. If we have a recessive R, which is an R allele, and a partial dominant R allele, this is 50%, so we are going to have a pink color. If we have in all R alleles or partially expressed R allele, we see that we have in 100% and we expression and we have a red color. But if we have a recessive and a recessive, which is now, there will be no pigment, so we are going to have a white flower. So there is also what we call codominant alleles. These are alleles. The alleles are different but both dominant. So let's say we have a blue allele for blue color and the allele for red color. So these are both dominant. So there's going to be 100% expression of this allele and 100% of this allele. So we are going to get a purple color, a mixture of red and blue. So we have what are called Punett squares, or we can call them checkerboards. So let's just have a look into the combination of the genotypes using this Punnett square. So the Punnett square, let's start with dominant and recessive alleles. So let's say we have a dominant wild type and a recessive wild type, the dominant wild type and the recessive wild type. And then we draw a box here. So this on the x-axis and the y-axis, these are the gametes. These are the gametes. So we have the R gametes and in a small letter R a dominant allele and a recessive allele, a dominant allele and a recessive allele. So this comes from the genotype which we have. You realize the genotype is heterozygous. So here we have RR, and here we start to combine here and here. We see the result. Here is going to R, small capital, heterozygous, here heterozygous, and here homozygous. So this, they are going to be red, and this is going to be white. So the ratio of the expression is going to be 1:3. These are one white and 3 red. So we will see that going on to the partial dominant. Just certain partial dominant. Let's say we have the dominant and the half and the partial expressed, and here we have the recessive and the partial expressed. So before we do we just explain this one, you will realize that this is a cross of a red flower and a red flower, and these are heterozygous, so that means they are carriers and they all produce a white flower. And here we have dominant and recessive and partial, dominant and partial, and partial and partial. So you realize that we have here a red flower, here we have a pink flower, here we have a red flower, here we have a red flower. So you realize that we also have a ratio of 1:3. That's one pink and one red and 3 red. So that means there is a relationship between, when you come to deciphering which is which, you have to go further to more crosses. So because we are having the same ratios here, although we are having the different colors. So going on further to codominance, let's say we have the blue allele and we have a red allele, a blue allele and a red allele. So this is a cross of two purple flowers. So here we have uh, BR, BR and RR. So we see that we have a blue here, blue flower. Here we have a purple flower. Here we have a purple flower. Here we have a red flower. You'll realize that you have a ratio of 1 is to 2 is to 1. This is for blue, purple, and red. So this is the phenotypic ratio which will be shown of. So this gives us a probability of the phenotype which are going to come out. For example, we can say the blue is 1 out of 1, 1, say 1/4, which is 0.25. And so the probability of having a blue color is 0.25, and that of having a red color is 0.25 again, and that of having a purple color is 0.5. So this is what you'll be seeing. So thank you for watching. Thank you.